Hi, everyone. Good morning or good afternoon, depending on where you are in the country or around the world. Welcome to week 3 of your journey into the world of the unseen. Now, the big takeaway from this week is that bacteria are, well, sneaky little boogers. I know that's not proper scientific verbiage, but the fact is, bacteria are, for the lack of a better phrase, sneaky little boogers. Over decades since the development of the very first antibiotic, that's penicillin, you know that, back in 1941, dozens of others have been developed, put on the market, and are currently in use in medical settings around the world. And yet, as brilliant as human science and medical researchers and engineers are, those sneaky little boogers, the bacteria, I mean, of course, still seem to devise ways of skirting around our human attempts to control them, destroy them, and minimize their damaging effects upon us. And once again, the most unsophisticated organisms on the planet seem to have the upper hand over the most sophisticated organisms on the planet. That's us. And it makes me wonder again, as you've heard me ask before, who really is in charge down here? Now, below the published announcement, I provided a link to a short video from Science News. And you will see how bacteria are able to develop resistance to antibiotics and confer that resistance onto their progeny really in very short order. Now, when you recall, the generation time of E. coli is about 20 minutes, you can appreciate how fast things can happen in the world of bacteria. And this week, as you culture bacteria in the exercises, the practical exercises, pay particular attention to how fast those sneaky little boogers are able to do their work. So this week, you're culturing bacteria using aseptic technique, and you'll learn about the effectiveness of various antibiotics and antiseptics, disinfectants, and controlling or inhibiting, I should say, and or inhibiting bacterial growth. Housekeeping items for the week. Now, you should have your microbiology equipment kit from Carolina Biological Supply by now. Open it up if you haven't already. If you put your media and your antibiotic discs in the refrigerator, get them out. You want to make sure that those items are at room temperature before you work with them. So, if you go to the course page in Connect, T3, topic 3, on the right side of that page, you will see a vertical, it says resources menu. This is not your class resources that I post things to. That's on the left side. But on the right side of that page, you will see a vertical menu. And if you scroll down through that menu, sort of near the bottom, you will see the streak plate and then another Kirby Bauer. That's the antibiotic disc activity you'll be doing this week. So you will see a little paragraph, couple of sentences on the streak plate, and then you'll see a little blue link, and then same thing with Kirby Bauer with the antibiotic disc exercise. You'll see a couple little sentences and then a little blue link. If you click on those blue links, it will take you to the Carolina Biological Supply page where you will find the instructions for doing both exercises, one for the streak plate, one for the Kirby Bauer. Now, the idea of the streak plate is what you will do there, you will mix parts, a little bit of each of the two bacteria cultures that you reconstitute. You'll mix them together, and then you will use your little plastic hoops, inoculating hoops, and you will streak that mixture 4 times onto one agar plate. And then when you each time you streak it, the explanation is there. But each time you streak it, you dilute the mixture. And in the fourth quadrant or in the fourth streak, once you grow the bacteria, you'll be able to see two separate colonies, pure colonies. And so we use a streak plate when you could have like 5 different cultures in there and you don't know what they are and you want to identify them. So how do you do that? We'll use a streak plate and you streak in the 4 quadrants, and in the last quadrant, you'll be able to pick out pure colonies of each of those 5 organisms on that from that streak plate. And then you subculture those in onto another agar dish or onto an agar slant, which you got in your kit, but you will not use them, but you could um, subculture each of those 5, in your case 2, cultures onto a new medium, incubate that, and then you have a whole bunch of pure organisms then you can identify and do other scientific things with, experiments with. So that's the idea of the streak plate. Anyway, go to the right side, scroll down, you'll see Carolina. You'll see the blue link to Carolina's instructions for the streak plate and also for the Kirby Bower. The first thing you need to do is reconstitute your organisms, your bacteria, and you got 2 little um, plastic or glass, I think they're glass now, little vials. It has a little stopper on top, and they have a label on there. And one is E. coli, and one is Strep aureus, and you see a little dried. It's a freeze dried chunk. In the bottom of each of those, you take some nutrient agar, squirt it in there, reconstitute it, rehydrate it, pour it back into one of your tubes, your um, with soy broth tubes, and then you incubate that. You do that for both cultures. You need to incubate these in a warm, dark environment. Optimum conditions for bacterial growth are warm, dark, and moist, and that's why the human body is such a good incubator for bacteria because we are warm, 98.6 or 37 degrees C inside, definitely very moist, and of course it's dark in there. You need to incubate in a warm, dark place. The moisture comes from the medium. And then you should see growth in 24 hours. If not, you culture for another 24 hours, but you should see growth in the first 24 hours. And you can tell there's growth in your um, tubes, right? Because they are, see how mine is kind of cloudy? Call that turbid. So you'll see growth in there. If the clear broth gets cloudy, then you have bacterial growth, and that's what you want. You should see that after 24 hours. And then you can draw from there and then incubate or then inoculate your agar dish and go from there. Okay. There are instructional videos in the Carolina Biological supplied instructions. So watch those little videos. If you have any questions, let me know right away. I made a couple of videos several years ago when I first started doing this class, and I can post those for you in class resources just to give you my take. They're old, but it's the same thing you'll see in the Carolina little videos. Carolina has redone their whole instructional packet over there. And so that's why I refer you to that, because they give you the latest information, and the videos are pretty well done. So shouldn't have any problem either reconstituting your bacteria or carrying out your streak plate and Kirby Bauer. So you're going to incubate the tubes, incubate them both at the same time in a warm, dark place. 24 hours, you should see turbidity or growth, and then you can do your streak plate and so you can have one streak plate and two Kirby Bauer, one Kirby Bauer plate for E. coli, one Kirby Bauer plate for Streptococcus with the little antibiotic discs on there. And then you're going to incubate those plates for another 24 hours. Incubate them. I usually, once I inoculate the plate, I put just some masking tape, just clear tape, whatever, just to keep the lid on so it doesn't fall off. You can do that, and you want to incubate these plates again in a warm, dark place, upside down, because what will happen is the medium, the agar, is moist, and so as the medium warms up, then some moisture will get released, and it will condense on the clear lid of the plate. And what you don't want, if you incubate them with the lid up, then the moisture will drop back down onto the cultures and contaminate your cultures. So you incubate your plates upside down for that reason. Okay, more on that as we get into the week. But you want to do that right away, because those reconstituted cultures, need to incubate for at least 24 hours to begin with, so that you can have growth bacterial cultures to do your other assignments or your other activities with. Okay, so you're going to make your initial discussion post. It's going to be no later than Friday, March 27th. Some of you have received a note from me that said, because some of you will wait until Friday to do that initial post, which of course you're going to get full credit for that. However, you'll get a note from me that says if you post earlier in the week, you can participate more fully in the discussion during the week. If you post on Friday night, that really doesn't give your peers enough time to read your post, do some minor research on the topic, and then substantially react or respond to your post. So just keep that in mind. Also, if you're responding to someone's post on Saturday or Sunday, don't put in follow-up questions. All right. Most people do their discussions during the week, because you have to do 2 posts on 3 different days during the week, and so you can't do all 6 posts on Saturday and Sunday. But lots of people like to do follow-up questions, like you respond to somebody and then you have a follow-up question for them. Don't add follow-up questions on Saturday or Sunday. People are done with discussion. They don't have time to research your question. They're not interested in responding to your question. So just be aware. It's okay to respond to somebody on Sunday. Just don't put in follow-up questions. That's just a ridiculous thing to do. Also, review my requirements for discussion posts. It's located on the left side class resources menu. Go to class resources and it says Jeffrey's guidelines for discussion posts. Read that, because some of you are not meeting the requirements for discussion posts. You need a minimum of 150 words, and you need at least one authoritative reference, properly formatted and properly cited within your text. Some of you have never done this before, And so that's why I posted the guidelines to give you an idea so that you can see how it's done in academia. So you want to make sure you do that. Make sure that you have 150 words, and your response is not, "Hey Joe, you did a great job. I really like the way you described this, and it may, you need, it's called substantive for a reason. You need to put something substantial in your post. You're responding to what your peer or what your colleague has posted, responding to that in an academic and in a substantive way. You're going to pick up on something that they said, maybe something that they put in their post, catches your thinking or your imagination. You do a little bit of research on that, and you respond to that, or you add to something that they've said. They'll respond to, they'll post on something. You'll say, "Hey, you can also." Um, in my research, I also found about this. Da da da. Here's the deal behind discussion. Think about it as though we are all in a classroom sitting around a big table, and I throw out a topic, say pathogenic E. coli, and everybody takes 10 minutes and does a little bit of research, and we go around the table, and Joe says this about pathogenic E. coli, and Sally says this about pathogenic E. coli, and then Bill says, "Hey, Sally, that's true, and this is what I can add to that." Okay, it's a discussion forum. It's not like kudos to your colleagues. It's not like you did a good job, and I like the way you did this, and it's you're not showering kudos onto your colleagues. You're adding to the discussion in a substantive way. That's why we call it substantive or substantive, depending on where you are. Okay, so just keep that in mind. Check out my guidelines for discussion posts this week. First week, I don't really, it doesn't really count because it's just introductory stuff. So I usually give everybody credit for everything on the first week. And then, but in the second week, I kind of am sort of hard on you because I want you to really understand what goes on in an academic discussion, and that's what this is. Okay, so I do count words. So just make sure you have at least 150 words. And if you look at my discussion, look at my guidelines, I do a post in there, and I think it comes out to be 155 words or something, not including your references, not including the, "Hey, Joe, I like the way, I appreciate," because I always put, you know, "Hey, Bill, I appreciate your thoughts on this week's discussion post," or, "I enjoyed reading your post." That doesn't count in the 150. It's the substantive stuff that counts in 150. Your references does not count. Okay, so just be aware of that. And if you do follow-up questions, that doesn't count in the 150. But if you look at my thing, look at my little post, I think it's like 155 words or something. And you can see it's quite substantive. And I put a note on there. I said, "See, look, you can do something that's academic, that's substantive, and that doesn't um, cost you, you don't have to sit for half an hour writing 5,000 words." My 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 always my thinking is, or my things that I tell my students is, if you just concentrate on writing a substantive post, then the 150 words will take care of itself. Okay, so there's that. All right. So initial discussion post Friday, March 27th. That's 11:59 p.m. GCU time, and your peer responses, all six of them, must be submitted no later than Sunday, March 29. And remember, you have to do two on the course of 3 different days during the week. Working on topic 3, methods of culture transfer, pure culture isolation, and antimicrobial testing. Pure culture isolation, that's the streak plate, and the antimicrobial testing, that's the Kirby Bauer. You want to complete your pre-labs for labs 3 and 11. Those are due no later than Sunday, March 29, 11:59 p.m. GCU time. There are two activities that I've described already. Start them if you haven't already. Some of you may have started yours over the weekend, which would be completely okay, but start them definitely today because they do need at least 24 hours to incubate. So make sure you get those in. If you have any questions, you're not sure if you have growth or not, just send me a message in Halo. Generally, I'll say, send me a photograph. Just use your phone, take a photograph of your tubes, and let me see them, and I can tell from your photograph if you have growth or not, substantive growth or not. So if you're not sure, drop me a note and send me a photo, and I'll let you know you're good to go, or you need to let it wait for another, let it incubate for another 24 hours. Okay. Let's see. Oh, and here's a caveat. So technically, both of those assignments, the streak plate and the Kirby Bower, are due technically no later than Sunday, March 29, 11:59 p.m. GCU time. That's this Sunday, March 29. However, I do not want you to rush through these exercises and just post something. Like, I don't want you to post something and then put a note on there. I didn't see any growth, but this is what I have, and here you go. If you need additional time, it's not a problem. I can extend you, okay? I don't want you to rush through and just post some garbage just to get the thing done by Sunday. If you need some time for extra time for bacterial growth or technique or whatever, let me know and I can extend you. It's not an issue. All right. And I don't charge for late submissions, so you don't have to worry about that. Okay. Um, I think that's all uh, that I have for the moment. I'll post a couple of little instructional videos as well as use the Carolina Biological Supply videos in the instructions because those are pretty good. If it seems like I'm a little out of it, I think I'm coming down with. There was a bunch of people with the flu in church yesterday. I think I may be coming down with something, but don't 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 worry about it. Just keep me in your prayers. Let me know what's going on. Have a great rest of the week. Oh, I have a class, another class that ended last night. So I have final grades to do for them. So I may be a little bit late. It might not be until tomorrow that I update your grade book. So you don't have to bombard me with messages there. I'll get to your grade books as soon as possible. Hopefully tonight, but we'll see. I'll do my best. Um, if not, certainly no later than tomorrow. If Wednesday rolls around and your grade book is not updated, you might want to check on me to make sure I'm still alive. Okay, that's all I have. I'll be back with you again sometime this week and have a great week.